So I'm very, very happy to introduce Dr. Bryan Kolb, our much admired neighbor of the Canadian Rocky Mountain. He is a native of Calgary and is currently an Emeritus Professor in the Department Neuroscience at the University of Lethbridge and never stopped to work, where he has been since 1976. It was not yesterday. His recent work has focused on the development of the prefrontal cortex and how neurons of the cerebral cortex change in response to various pre- and postnatal development factors, including hormones, experience, stress, psychoactive drugs, neurotrophins, noise, and injury, and how these changes are related to adult behavior in laboratory animals and children. Bryan Kolb has published 7 books, including 2 textbooks with Ian Wishaw, Fundamentals of Human Neuropsychology. Now it's in its 8th edition. It's a must. Introduction to Brain and Behavior, now in 7th edition, and over 450 articles and chapters. Bryan Kolb is a fellow of the Royal Society of Canada and an officer of the Order of Canada. He has 4 honorary degrees, UBC, Concordia, Thompson River, Lethbridge, and is past president of the Canadian Society for Brain, Behavior and Cognitive Science, past president of the Experimental Division of the Canadian Psychology Association, and past co-director of the CFAR program in child brain development.
Professor Bryan Kolb will lecture about stress and cortical plasticity in the developing brain. Welcome, Dr. Bryan Kolb.
Thank you very much. It's a pleasure to be here, and I'll try screen sharing here. There we go. Okay, so I want to thank the previous speakers for introducing some concepts that I'm going to show you. One of them is epigenetics, and one of them is skin-to-skin contact, which is great. The organizers did not know that was going to happen, and neither did I. So that's great. Okay, so if we look at the take-home message that I'm going to leave you with, it's that experiences that are both preconceptual, which we haven't heard about before, life begins at conception, but the effects on life predate that, prenatal and early postnatal stress. Early intervention, I'm going to show you, can actually reverse many of these brain changes. So if we look at the types of developmental stressors, preconceptual, maternal, and paternal. Now, a lot of people are going to say, what's a dad got to do with it? Turns out a lot. The effects of preconceptual experiences by the dad have a bigger impact on the baby than the mom, and I'll show you why. Not to say the mom doesn't. Prenatal, so we can look at things like maternal mental health, which was alluded to by Dr. Verney, and also other stimulations. One we've used experimentally is noise. Noise is a big stressor, and one source of noise is traffic noise, and we've used that in our animal studies to stress pregnant moms, as you'll see. And then we have postnatal stressors, which I won't talk about, but you can see all kinds of stuff. So given the time constraints, I'll just focus on the first 2. Okay, brain plasticity. What is this all about? The idea is that experience changes the brain. It's changing your brain right now. So if you learn anything from these lectures you're hearing, the speakers, including me, have managed to get into your brain and change it. Your brain is plastic, and with any luck, you'll remember some of it. So how does it do this? This gets to epigenetics. It's altering gene expression. So you have, as you heard from Dr. Verney, you've got nature, which is genes, you've got experience, which is nurture. The junction of these is epigenetics. So what does that mean? You have something in the order of 24,000 genes. Every cell in your body has those genes, but those cells are different. The cells that make up your skin, your eye, your bones, and so on are different. How is that? It's because, and I'll show you in a second, the genes can be turned on or turned off. So genes can be turned on or off by quite a number of internal events, but also experience can turn genes on or off. That is the idea behind epigenetics. So if we have genes being turned on or off by experiences, this leads to brain plasticity. So this is what's driving the change in the structure of the brain, and that leads to altered behaviors. I hope you get a better idea now of what this epigenetics is about, and I'll give you a concrete example. So what does prenatal stress do to the brain? None of it's very good. It alters the volumes of specific regions in the brain. It alters functional connectivity, that is the connections between different regions and among different regions, and it also alters neuronal morphology. I'll give you some laboratory and human examples. Now, the human example I'm going to give comes from workers at McGill, and it's one of the most extreme examples of stress, but it does illustrate the point. So, in Quebec in 1988, one of the worst natural disasters in Canadian recorded history occurred. The power went off for an ice storm for up to 6 weeks in the coldest month of the year. That was a very cold year. It was about -20. So people could leave and go to Toronto or somewhere else where they did not have the stress of being in this disaster, or they were stuck there. And so there were women who were pregnant at various stages of pregnancy, and some who were not yet pregnant. So we have preconceptual events as well. And so we can actually, the people at McGill can actually study the effects that this stress has had on their offspring. The children have been now followed for almost 20 years. I realize that 1988 is more than 20 years after the event, but the most recent papers are studying the kids who are now 20. Okay, if you don't know what the ice storm was like, this was it destroyed the electrical grid. You can see this, and you can see here the effects, and you can see a lady walking her baby in a buggy there by these ice-covered cars and benches and whatnot. So what did this do? Now, don't freak out over this figure. It's not complicated. It just looks like it would be. This is gene expression. So genes that are green are not on very much. Green genes that are red or orange are. So what's this? This is the retrospective level of stress that the mums had. These did not go to Toronto or somewhere else. They stayed in Quebec. And these are women who were not stressed. They were not there. These are individual children going across this way. So each row is a child, and these are different genes. And I think what you can see here, I hope, is that if you look at the stressed kids or the stressed mums, there's a lot of red here. So these genes are turned on. And if you look at these ones are not. Okay, but now let's look over way over here. We can look and see that these genes are turned off. And down here, these ones are turned on. The pattern of gene expression is completely different. Okay, in the kids in the ice storm and the control group. So what's that mean? It means that prenatal experience is altering gene expression and thus brain organization and behavior. Okay, so we're going to expect, if we investigate the brains of these kids, that they're going to be different, and we're going to see that they are. And the behavior is different because they were in that previous group. Okay, so what's this mean? It means that prenatal experience alters gene expression and therefore brain organization. But you noticed that not every kid was the same. There's variability in the pattern of altered gene expression. Why is that? You would think that if it was a really nasty event, they'd all behave the same way, except genes don't have one form. We have alleles, and so different genes have different alleles. So we have here 3 different alleles of these genes, and you can see that depending on which allele they have, the size of the hippocampus is different. So while I can tell you that the hippocampi of the kids who as you'll see, who were in the ice storm group it's actually bigger than the control group. Their hippocampus is actually larger. How much larger it is depends on which allele of the gene that they have. So they're not all going to be the same. Okay? So the severity of prenatal experiences depends upon individual differences. So we're not going to be able to say, oh, all the kids, or all the children who are severely stressed are going to show this kind of behavior, and the ones that didn't, different kind. No, that's not quite right. There's going to be a lot of differences among both groups. Okay, we can do similar studies in our lab animals. And so, I'll do this first. So if we look at what the McGill people have found is that 3 regions, hippocampus, amygdala, and prefrontal cortex. Prefrontal cortex is the stuff here. It's a stupid term. Pre means in front of, right? It would mean in front of here. Well, in front of front, but that's not what it is. It's a region of the frontal lobe. So prefrontal cortex, amygdala, and hippocampus are all unusually large. Not the effect you might expect. However, they're not connected properly. How can you study this? You can simply put any one of us in an MRI and say, don't think about anything particular, just lie still. And then we can record brain activity, and it's called resting state connectivity. This gives us a measure of a variety of things, and I won't bore you with all the things you can measure. The point is that you can look at the connectivity, and when you do, what you find is that the reduced resting state connectivity is found in the kids who have the larger structures. They have larger structures, but they're not connected properly. This means there's a mechanism here for changing brain function. Over this. This is mice. And we're looking here at activity. So here's control mice. And here we're giving the mouse a sound. So here, when we start it, there's nothing. And over time, over the first milliseconds, you can start to see activity. This is just in the left hemisphere. You can start to see activity appearing here and getting stronger and stronger. The redder it is, the stronger the activity. Let's look at the mice who have prenatal stress. They don't show that. They show activity, but it's way reduced. And we can look at other, here, stimulate the paw. So we're going to poke the paw, which is going to do the same thing. It's going to, this is now the activity before you start poking. There's not much, and over time you get more and more and more. And again, if you look at the mice who had prenatal stress, this was sound. It was in the equivalent of the second trimester of humans. It was about 80 dB intermittently for about 12 hours a day. And the sound was Tehran traffic noise. And the reason it was Tehran traffic noise is because our postdoc was from Tehran, and she decided she would use that because she hated it. So there you go. So you can see that even in the mice, we can see prenatal stress has altered brain connectivity, just like it did in the kids in the ice storm. We can also look at structure. So and the advantage we have in the animal studies is that we can actually look at the structure of cells. So here are cells in, this is hippocampus. This is in a region called nucleus accumbens, which was alluded to in the previous talk as a pleasure center. This is prefrontal cortex, so is this. This is parietal cortex. The cells aren't all the same, but the key thing here is if you look at an example of a brain that had no stress, and this is a hippocampal neuron, and one that was stressed, you can see they're not the same. The stressed one is really reduced. So, what about preconceptual stress? Well, one of the ways we can look at this is at the stress that a dad, potential dad or mom will experience prior to conception. So let's look at dads. If we look at dads, we know that the sperm is being reborn. We're making new sperm all the time. Mothers are not making new eggs. This is going to be one of the reasons why the effects are bigger from dads. So if you have a dad who is, for example, a heavy drinker, preconceptually, the gene expression in the sperm is going to be different than if he was not doing that. In our animal experience, we stress the animals. I'll show you how. It's not nasty, or whether they're dads or moms. And what we do is we look at the change in gene expression in sperm and how that affects gene expression in the offspring, okay? So here's a potential dad. He's sitting on this little platform. Now, a rat is a prey species, and so it doesn't like being sitting out in the open because when's the cat going to come? When's the owl going to arrive? And the mums or the dads had to do this, or they were placed on that platform for 30 minutes twice a day for about 30 days. Okay? We can then measure behavior, and you can see a variety of behaviors that change. So one of the things we can see is that the babies, when they're adults, whose dad was on that platform, they haven't been conceived yet, have increased anxiety, they're not going to come out in the open, they have motor deficits. We can train rats to reach through bars and get food. They're really good at it. They have really dexterous fingers just like ours. They're impaired. We can look at play behavior, and the play behavior is altered. Play is really important, and it's a whole other topic. Play changes the structure of the brain. Not playing is a bad thing, and think of COVID. And you can just think about that for a second, and the absence of play in children. And finally, we look at learning, and you can see that animals are impaired at a variety of learning tasks. Okay. What we've got here, there's a neuron here, and you can see in the neuron that it has the structure you saw in the previous ones, but in addition, you can now see that there are spines, these little protruding things, and that's where the connections are made from cell to cell. So the number of spines, and we can estimate the number, and I won't bore you with how, but we can get a pretty accurate estimate of the number of spines tells us how many connections there are. So we can predict that in the ice storm kids, the number of connections is reduced, and we see that in the MRI, the resting MRI. So if you look here, this is the mean spine density in controls versus paternal stress. And you can see we have a significant drop in spine density, which means there are fewer synapses. Okay? Well, this is great. We can make animals worse, and my wife is a veterinarian, and she likes to say that one thing we're really good at is making our lab animals worse. Why can't you make them better? Well, we can, and we just don't do it as often. I've been studying brain development for about 50 years, and so it's only been in the last 20 years that we've started to see if we can't reverse some effects. So what are we going to do here? Tactile stimulation. Dr. Buckley introduced us to this, and that was nice. Skin to skin especially is important. So we're doing work with the neonatologists at the University of Calgary, and the idea is that when babies have a perinatal insult, what's the treatment? Good luck is the treatment. Well, no, the thing to do is to start with tactile skin-to-skin contact. In our rats, we have a little brush for rats, and we just brush them. We line them up like little Vienna sausages, and we brush them. And 15 minutes, 3 times a day. Now, the neat thing here is that they go into dream sleep. They go into REM sleep. So this is not stressful. In fact, it seems to be a positive thing. Mice are a little different. This is one of our former students, and he's tactily stimulating this mouth with his finger. Believe it or not, the mice love it. So this is a young mouse, and he's just doing this with them postnatally. It's a little older than this baby rat. Okay, both of these have huge impacts on the brain. I can't get into all the details, but how does this happen? So Dr. Buckley introduced the idea that tactile stimulation is changing the brain. She suggested it through oxytocin. I'm sure that's partly true. There's probably other routes too. There are 2 kinds of nerve fibers from the skin. There are fast nerve fibers that make you move away. There are slow acting nerve fibers that are not myelinated. They don't have insulation on them. Here's a feather as an example. You don't move away. And in fact, in Dr. Buckley's talk, the baby's grabbing onto the mum, right? It's positive. So we know that these slow nerve fibers are stimulated by the tactile stimulation. And that makes a difference. It's altering the brain in a different way than the fast nerve fibers are going to be doing that. It's a route into the brain and alters brain structure and function for good. Okay, so what can we conclude from all this? Well, brain development is complex and is prolonged. In humans, brain development continues to about age 30 years. It's a long process, but the key times are prenatal, perinatal, and adolescence. And we've only talked about really the prenatal in this presentation. Its influence, plasticity, is influenced by a whole range of experiences that are both pre and postnatal. So we can have the preconception ones, and we can have the postnatal ones. And finally, we need to identify procedures or things that can reduce the stress both pre and postnatally. And in my last minute, I'll give you an example here. We have a mouse model of Alzheimer's disease. What's this got to do with babies? Everything. So we have the babies, because of the altered genes that were inserted into the mice, will cause them to start developing Alzheimer's symptoms, so bad memory in particular, but plaques and tangles, in plaques anyway, in the brain at about 4 months of age, quite really early because they're going to live 2 years. And so what we did was to say, okay, what if we take these mice, and we can also give them the noise in utero, and we start tactily stimulating them as babies. Does this affect the progression of Alzheimer's in adulthood? Well, it stopped it. It blocked the Alzheimer's changes. Now, you could say, well, when did you do that? Well, we looked at them. The latest we looked at them was about 9 or 10 months. It may be that they would start showing up even later. We haven't done that to this point, but it, in fact, could be the case. We also wondered whether we could initiate the tactile stimulation later. Do you have to do it when they're babies, or can you do it when they're a little older? You can do it when they're older, when they're adolescents. It just isn't quite as effective. Okay, so tactile stimulation is one way. Another way that was introduced also by Dr. Verney was the interaction between the mom and the offspring. Okay, this physical interaction, the mental interaction, and whatnot. Okay, I'll stop there, and I just want to thank my colleagues. In particular, I want to thank the late Fraser Mustard, who got me onto a lot of this. He was at McMaster. He started McMaster Medical School, but he was the person who really got us thinking about how early experiences gets under the skin to change the brain. Okay, thank you. I can unshare.
Thank you very much. So we have a few questions about what you said. So Susana Lopez asked, when we have a stressed father, the role of a calm mother becomes even more important for the positive development of our baby? Is there a relationship when we have like a stressed father? What is a pregnant mother can shock absorb or transmute or transform the stress that she gets from the father's side?
Yeah, so we've done studies which we've called bystander stress. And what we can do is we can stress the father using the procedure that you saw, or we can stress a female who's also a pregnant female, and we can see that the gene expression and the behavior of the mom who's not stressed directly is changed, and that change is transferred to their offspring. So yes, the answer is that the stressful experience of the father can be transferred to the mom, when we call that bystander stress, and produces a different pattern of gene expression, and it's different in different structures, but it's there.
So, and in the same way, before the last question of Sarah Berkeley, I wish to ask you, we have this talk a bit previously, but what do you think about the time the father starts to impregnate his sperm to impact the baby? How many months before he should be sober, he should have a better life, he should, make a better epigenetics to have a better material to offer to his offspring?
In humans, the entire sperm bank within the male is replaced in about 40 days. So I would think that you're looking at, I tell my students, ladies, if you're thinking of having a baby, make sure that your partner has been good for 2 months, because that should, in fact, clean out a lot of that crummy sperm. We have done studies to say, okay, what if you do the following? What if you have a baby, and this is now in our rats, from a dad who was stressed, and then you wait and have the dad have another baby later, but the stress is all gone. Will there be a difference in the performance of the babies in the two groups? This is work being done by some of our graduate students. It's not completed, but the prediction is that the outcome will be different. That the dad who's not been stressed recently the babies will have a different pattern of behavior and gene expression.
That's fascinating because before what you bring today, the typical answer that we have of the cycle of a man was 1 week, that every week we change all our sperms, but it appears that in fact it's 40 days, which is quite important information for us today. There is Sarah Berkeley who say that, thank you, fascinating talk, and they have a severe flood in 2011 in Brisbane, and local researchers follows up women who were pregnant at that time. They found that having midwifery care reduced the neurodevelopment effect on children at 6 months. So just to share that there have been a research made at the Brisbane, a bit like the ice storm in 1908.
Well, they're doing similar studies with other disasters as well around the world now. One of them being the Fort Mac fire 6 or 8 years ago, whenever it was. Same issue. Yeah.
Vanessa asked if abstinence alone will clean up the sperm. Any research about other method of detox?
Well, the only studies that I'm aware of are in rodents. And in the rodents, it does seem to change the pattern of gene expression in the sperm. I'm going to go out on a limb and say that's almost certainly true in us as well. One of the differences is when you're doing animal studies, you have total control over the experiences in people. And so while they may have stopped drinking, they may have started doing something else, which is going to confound it a bit. But I think the idea, at least in the animal studies, is yeah, it does work.
So it does work to have it's exactly the study that your student is doing in a moment. When there is an abstinence of alcohol or bad behaviors or negativity, it clean up and we start fresh. Okay. Well, you bring amazing news today, and it's opened the part of the father which is most of the time diminished, and see that the responsibility of our offspring really relate on both gender. Thank you so much.
Thank you so much
for your amazing work.